Hereby I open this academic ceremony in which Ioannis Lempesis will defend the academic thesis, Body Fat Distribution and Obesity, a comparison of upper and lower body adipose tissue biology in humans. May I invite you to present a summary of your study and the conclusions of your thesis.
Dear Mr. Director, thank you very much. And dear members of the assessment committee, dear friends and colleagues and other attendees, it's a great honor for me today to be here and present the main outcomes of my PhD work that resulted to my PhD thesis entitled Body Fat Distribution and Obesity, a comparison of upper and lower body adipose tissue biology in humans. This was a project between the University of Birmingham and in the United Kingdom and Maastricht University here in the Netherlands, and also a result of my interest to want to better understand metabolic diseases, and in particular obesity. Obesity, as we know, it's a chronic complex disease that is characterized by an increased accumulation of fat in our bodies, which at the same time poses a risk in our health. A more solid definition of obesity can be determined by using the body mass index, which is usually defined by dividing the body weight of an individual expressed in kilograms by the height expressed in meters square. And then, based on that BMI, we have three main categories of individuals with normal weight, individuals with overweight, and those with obesity, which is usually defined as equal or more than 30. As obesity is a global health problem and it's continuously increasing, people with obesity, people that are affected with obesity are more and more, and we can see here that over the past decades in this figure, women and men that are living with obesity are increasing, the number of them, and currently adults in Europe that live with overweight are more than 50% of the adult population, and approximately 15% of the adults live with obesity. As seen before, increased fat mass is a characteristic of obesity, and it's important as this is linked with increased health risk, as it's increasing the risk of developing many diseases, including type 2 diabetes, what is commonly known as high blood sugars, cardiovascular diseases like heart attack and stroke, and many types of cancer, imposing and underlying how important is obesity on itself as a health problem, but also in relation with the comorbidities. Fat tissue is a very important organ, and it's changing in obesity. It's very important as it is taking part in many processes in our body, and it's important for the whole body physiology as it regulates glucose and fat metabolism. And as it is changing in obesity and becoming dysfunctional, many characteristics and properties of the tissue are changing. Of those include the increase of the fat cell size, which in turn is linked to an increase of inflammatory factors and changes in adipokines. Adipokines are a general term that describes the hormones, proteins, and the inflammatory factors, among others, that they are produced typically from fat tissue. Another thing that is changing in obesity, and it's very important for the physiology of every tissue, is blood flow. In most individuals with obesity, it's recorded that blood flow is reduced, and this is affecting the function of the tissue as blood flow supply. It's an indicator of the supply of oxygen and the nutrients to the tissue, but also of the circulation outside of the tissue of the hormones and the nutrients when needed. Another thing that is changing in obesity is the mitochondrial function. Mitochondria represent the powerhouses of our cells that consume oxygen to produce energy, and it seems that they are not working again properly in obesity. Another thing that is also changing is the oxygen levels. Oxygen levels are a delicate balance between the oxygen supply, as expressed with blood flow, and oxygen consumption, as usually predominantly expressed with mitochondrial function. And I will come back in a few slides to this. Another thing, apart from the increased fat mass in obesity and the link to disease development, it's important to think of the different body fat distribution patterns, meaning where the fat is stored in our body, as this is also linked to a differential, meaning different disease risk. Abdominal fat for accumulation, the belly fat, upper body fat, is linked with an increased disease risk of cardiovascular and diabetes. However, the lower body fat, femoral in the thighs and gluteal in the hips, seems to be protective. However, we are not fully aware of the differences in between these two depots. And in my PhD, among the objectives and the aims, it was that we wanted to better understand this difference between these two fat depots. The first objective, it was that we wanted to develop a technique to measure fat tissue blood flow. As said, blood flow is important. Usually is measured with a gold standard technique that we want also to utilize in my studies, the xenon washout technique that use a radioisotope, 133 xenon. However, it was not feasible to use it early in my study, so we have to come up with another solution and to develop a technique. The second aim and objective was to compare the adipokine production and inflammatory factors in the two fat depots, in the upper and lower body fat. Again, comparing these two fat depots, we wanted to look into the differences when it comes to mitochondrial function. And the final objective of this PhD project was to examine the effects of the different oxygen levels on the production of adipokines and inflammation in fat cells in the laboratory. To achieve these objectives, I was responsible and lead two studies and recruited healthy volunteers for the first study to examine the Doppler ultrasound as a novel way of measuring adipose tissue blood flow. 17 healthy individuals were recruited, males and females. And for the second part, the second study, where we wanted to look into the physiology in humans, but also the experiments in the material that was taken from these volunteers in the lab. 23 postmenopausal women with other normal weight or obesity were included. As part of the human studies, in both studies, I have examined the adipose tissue using the Doppler ultrasound to measure blood flow. And also on the second study, blood samples were taken, 3 blood samples at any time point, because we wanted, of course, from the fat tissue depots, specific tissue blood samples, but also arterial blood samples, because in particular from the radial artery, as arterial supply give us an idea of how much of a substance of interest, for instance, oxygen, goes into the tissue. And then with, at the same time, we were taking blood samples from the small veins that particularly drain fat tissue, and we had a tissue-specific venous blood sample to have an idea of how much of the substance goes out of the tissue, so we could calculate how much oxygen could be used from the tissue, how much carbon dioxide could be produced, and the same for the adipokines of interest. The next thing that was important in the methodology, it was that we have taken fat tissue samples from these individuals, biopsies, and from the two fat depots, and one part of them was used to determine fat cell size, another part was used for determining the expression of adipokines, markers of inflammation and oxidative metabolism. Another important part, it was to isolate fat cells and culture them, and when they were mature fat cells, to execute experiments, measure the same adipokines and markers. Next, to execute dynamic experiments with these fat cells to determine the mitochondrial function, how much oxygen the cells are consuming. And the last set of experiments, that it was based on equipment that give us the opportunity to regulate, to choose in how much oxygen the cells live inside, to see how different oxygen levels affect the production of adipokines and inflammatory markers. And this leads me to the results of my studies. The first objective, to measure blood flow using the ultrasound Doppler technique, as we can see here, that was indeed feasible. Starting from the left hand of the figure, we can see that blood flow in fasting condition after 10 hours of not eating, that we had significantly higher in the abdominal fat depot as opposed to femoral, which it has been seen also in previous studies with the gold standard technique. And then after these participants have drunk a glucose drink, we see an expected increase in abdominal fat depot of the blood flow following 2 hours after that drink. Next, this set of results comes from the experiments combining the human studies, the biopsy results, and the fat cells experiments in the laboratory. Here we can see that the interest goes to examine adipokines and inflammatory markers. When it comes to comparisons in the in vivo physiology study, MCP1, which represents a pro-inflammatory adipokine, was highly released from abdominal fat depot as opposed to the femoral fat depot. When we examine other adipokines, we could not see any significant differences in between the two fat depots. And the same for this group of individuals, it was true that we could not detect significant differences in the blood flow between the two fat depots with this technique. Next, in the biopsies, despite not finding significant differences in the fat cell size between the two fat depots, we have shown that the expression of pro-inflammatory adipokines in the femoral biopsies, the tissue comes from the femoral fat depot, was higher for several adipokines. And the final part of the experiments, when examining fat cells, which represent one of the cells that they exist in fat tissue, we have seen that several pro-inflammatory adipokines were produced in higher levels in the fat cells deriving from the abdominal fat depot. In the next part, in the same set of experiments that we were interested in examining what we call oxidative signatures, the mitochondrial function, and how much oxygen fat tissue is using and consuming. Here we can see again the same setting with the in vivo physiology studies, biopsies, and the fat cell experiments. Despite no differences in the blood flow, as also seen before, between the fat depots, we were able to show a significantly higher oxygen extraction in the femoral fat depot for these individuals as compared to the abdominal. And then we examined in the biopsies several mitochondrial markers, but we were not able to detect any differences between the two fat depots. Next, we examined also these markers in the fat cells deriving from both fat depots, and still we did not detect significant differences. However, when we did execute the dynamic experiments and we have examined these cells in the laboratory, we were able to show a significantly higher oxygen consumption rate for the adipocytes that they were deriving from the femoral fat depot as opposed to the fat cells from the abdominal, and that was in line with the observations in the human study. The last set of experiments, it was about to examine the effects of the different oxygen levels on the production of adipokines in the fat cells, again from individuals with either normal weight or with obesity in the abdominal or the femoral fat depot. As this specific equipment gives us the ability to choose in which oxygen levels the cells live inside, for 2 weeks we have used the standard lab conditions, which are the 21% of oxygen, the air we also breathe now. And then, based on measurements that they have been executed in humans, and we know that in living condition, fat tissue levels, the physiological levels are between 3 to 11%. We have chosen the average of the high physiological levels for the 10% of oxygen, and then the average for the lower physiological levels to be the 5% of oxygen, and use them for the cells. When we compare for the cells deriving from the individuals with normal weight, we have seen that from 21 to 5%, an increase in leptin, in release and production, that in expression and release, that it's in line with previous observation because leptin is regulated by oxygen, but no significant changes in other adipokines. Next, when we compared the cells deriving from individuals with obesity, in 10 and 5%, what was interesting, it was again that we have seen leptin higher in 5, but a decrease of several pro-inflammatory adipokines at low physiological level, 5% as opposed to 10%. And this leads me to the main conclusion of these PhD studies. The first one, it was that indeed the measurement of the upper and lower body fat tissue blood flow is feasible with the Doppler ultrasound technique. The second, that the upper and lower body fat tissue are characterized by distinct inflammatory signatures. The third one, that the oxidative machinery, the mitochondria, seems to be functionally impaired in the upper as opposed to the lower body fat tissue. And the last one, that the oxygen levels impact the production and the secretion of the adipokines in fat cells from individuals with obesity, but not with normal weight. And at that point, I would like to thank you very much for your attention, and I would like to give back the word to Mr. Prorector. Thank you.
Thank you for your presentation. The opposition will be opened by Professor van Marken-Lichtenbelt. He is Professor of Ecological Energetics and Health at Maastricht University. Professor van Marken-Lichtenbelt was chair of the assessment committee, and he is the secretary of this degree committee.
Thank you. Dear candidate, first of all, congratulations with a very nice piece of work. It's very well written, nice to read it, and also contains a lot of different studies, from physiology to cell biology, from methodology to intervention studies. So interesting. And I'd like to congratulate also the promotion team with that. But now the serious part. First, a general question. Of course, there are different depots in the body, fat depots in the body. And as you may know, I studied brown fat, and then people thought, well, you should have as much brown fat as possible, at least more brown fat, and so. And now I read in your thesis, you state on page 8 that the femoral fat possesses protective function, properties, and inverses cardiovascular disease risk. So it's very positive. And now my question is, could you state the fatter, the better?
Dear highly esteemed opponent, thank you very much, first of all, for the kind words and for the quality of the PhD studies, and in particular, the points that you have mentioned for the thesis. I could say that there is not let's say, a strict answer, and to say that there's only one way of saying that. For instance, sorry, let me to start again. It's that definitely we know that BMI per se, it's an indicator of negative disease. So if we believe that BMI represents to an extent adiposity, the more fat tissue, based on epidemiological studies, we know that this is something negative. And usually we have in both fat depots this accumulation of fat, maybe in favor on one or the other. Based on this observation, I would think that potentially somebody to possess a greater amount of fat could be detrimental for health.
Okay, so but yeah, there must be some limit somewhere, I guess. So I think it's more the relative amount of fat and the distribution that matters, and maybe not the absolute amounts. But let's go to my next question, which is more about the techniques, the laser Doppler ultrasound technique. I think this is indeed very nice that you tested the technique. It's also very promising, but there are also some drawbacks, I think. And just a detailed question, because normally it measures then the blood flow, but as I know from my experiences with skin blood flow measurement, with laser Doppler flowmetry, that it gives you arbitrary units. But in this thesis, I see absolute. So the graph you showed, they are absolute in milliliters. I don't know what was it? Page. Milliliters per minute, yes. So that's an absolute value. So can you explain that? How is that possible?
Could you repeat the part of the question? I could not
understand.
Well,
normally what I know is that it gives you arbitrary units, or in other cases in relative units compared to maybe maximal blood flow. But you give absolute values, milliliter per minute.
Yes, for instance, this is indeed, if we compare it with a xenon washout technique, which is usually utilized for wet adipose tissue studies, this is also expressed per mass per unit of tissue. And in this study, because we have measured different also vessels that they are usually smaller, that they are measured with other techniques, they represent, in this particular study, bigger vessels. So I was measuring the volume indeed within the lumen per minute. So there is indeed a difference between the previously reported units when it comes to tissue. We didn't have also the ability to measure, at least in chapter 3, the volume, and we didn't.
But do you really think it's in absolute values? You really think you measure blood flow in milliliters per minute?
This is usually also how it's measured for other studies when they utilize Doppler ultrasound.
Or is it the instrument that just gives you that number?
The instrument gives you this.
And is it the dual beam, or how's it called? Dual beam Doppler system or single?
Actually,
the system you use, is it dual beam, or single beam? Because I understood that with dual beam, you may get a correction for the values you obtain, and you can correct for the angle of measurement, and with that, you get a better estimate of the actual flow.
There are some corrections that are done from the algorithm, I mean, from the machine, but when it comes for the angle, that description, it was about to try to avoid to be very closer to 19 degrees, but to be less, because it's a limitation of ultrasound anyhow, that it cannot produce signal or detect the signal.
Okay. Then may I interrupt, because my time is going. I want to ask one more question, very specific to this technique, that I really like to know the answer, because I think you search for the right vessels, that you get enough signal in the tissue.
Correct.
But the tissue may have different properties and different kind of vessels with size and diameter, etc. So is the data that means that the data not necessarily gives you changes in or differences in blood flow, but maybe difference in anatomical properties. What do you think of that?
This could be partially true, but when we select a particular vein, this is where we do the measurements for the same studies. So I think that we could not, if this technique was not good enough, let's say, to detect this difference, we could not see them following in so many participants. Yeah. I think that indeed it's able to detect some changes, some difference that they are there.
Okay, yes, I'm sure you did your utmost best to make the good comparison. Thank you very much for your answers. I'm quite satisfied, and I give the word back.
Thank you. The opposition will be continued by Dr. Tarani. Dr. Tarani is Honorary Professor of Medicine at the University of Birmingham. He was member of the Assessment Committee, and the university highly appreciate your participation to this academic ceremony.
Thank you. Very nice presentation, Ioannis, and congratulations on the thesis, and the very nice presentations, and congratulations to the supervisors. Oh, I can't take this one. Okay.
Please use your microphone. Thank you.
Yeah, thank you.
No microphone.
There's no microphone there, so I'm using this one. I'm borrowing the microphone. So just two questions I've got in mind. The first one is, you've shown very nicely how the different level of oxygens can affect the adipokines differentially between people with obesity and thin people. You also showed many differences between abdominal or upper body and lower body fat. My question here is, you chose healthy people, or even people with obesity who don't have complications. If we have a hypoxic condition, especially obstructive sleep apnea, which is very common in people living with obesity, how do you think this could impact your results? And do you think the impact will be differential between upper body and lower body, or will it be just universal same effect.
Dear esteemed opponent, thank you very much also for your kind words and highlighting the importance of the work. Indeed, we have, by definition, the participants that they have taken, that have been recruited in this study were considerably healthier than somebody would expect, also for the ones with obesity. And I think also there have been a few studies that they have examined that, and they have shown that moderate obesity, or what could be claimed as metabolically healthy obesity, represent a fraction of the individuals with obesity, and in them, potentially, we could see what we have seen, higher oxygen levels. However, it has been shown in particular for people that they have also obstructive sleep apnea or hypoventilation related to obesity, that there we have lower levels of oxygen in the fat tissue. So, indeed, still the results, they are not the same. Maybe depends on the population, on the disease stage, and comorbidities that they result to these differences. Now, if there could be potential differences in between the two fat depots, it could be claimed that maybe these hypoxic conditions in the total body, they change proportionally. And I believe that could be like that if they change in the same direction. And
thank you. The second question is related to the metabolic healthy phenotype that you've just mentioned. Do you feel that mitochondrial dysfunction is part of the reason why people with metabolic healthy obesity could move into the metabolic unhealthy obesity category? And what do you think might be the driver for that metabolic mitochondrial dysfunction, if it is a contributing factor?
So yes, indeed, this could be, if we think as obesity as a natural history of a disease that develops, it could be that indeed we see a defect in the mitochondrial function, affecting mitochondria. Mitochondrion is a very central organelle which regulates also inflammation and lipid metabolism, among other things. If we think that while becoming more dysfunctional, this could lead to a greater inflammatory status in general, and maybe ectopic fat deposition that drives the transition from the metabolically healthy to less healthy obesity.
But do you think there are any particular drivers might drive that mitochondrial dysfunction? What causes it?
It could be. I could speculate maybe of two things. Maybe there is an inherent tendency for these cells to be dysfunctional in those people that they are going to live in an environment with surplus of calories, so they have to develop obesity, but also to be a dynamic process of, for instance, crosstalk between inflammatory cells that infiltrate in the fat tissue during the development of obesity, signals that they compromising more the mitochondrial function. So it could be these two things.
Thank you for your answer. I'm very satisfied. Thank you.
Thank you. The opposition will be continued by Professor Keyer, Professor of Human and Animal Physiology at Wageningen University. Professor Keyer was member of the assessment committee, and also for you, thank you for joining and participating to this ceremony.
Thank you, Mr. Rector. Dear candidate, it was a pleasure to read your thesis. I understood that you, due to the COVID pandemic, had some problems in executing everything that you wanted. But nevertheless, a very nice thesis to read. Of course, I have some questions, and I will start with maybe two very general questions. And one is so you, in the study, in the first study where you tested the Doppler sound, you say that you have healthy individuals, but one is obese. And I thought that obesity was a disease. So I'm a bit puzzled. Are the people healthy or are they not healthy? So,
dear highly esteemed opponent, thank you very much for the kind words and for the question. So yes, indeed, so if I understand correct, you ask me if we believe that these fraction of individuals that we include in the second study that they are considered to be healthier even though they have a disease. No.
But by definition, I think obesity is a disease. It's classified as such by the WHO.
Yes.
Nevertheless, you say, okay, I have healthy individuals, and now you also say healthy obesity. But if it is a disease, healthy obesity doesn't exist. So could you clarify
Yes,
this?
That's true. That's true. Indeed, it's a bit conflicting, let's say, in a way that there may be an umbrella of definition that there is no just one obesity, but many obesities, depending on the body fat distribution and depending maybe on the stage that we detect an individual that has not yet developed cardiovascular disease or other complication, because even those that we call them, which is there is no unanimous definition for them, or if they indeed exist, the individual with healthy obesity, that a few years of follow-up, they will develop these complications. It depends maybe on each person.
So in essence, you say, for me, it's not the obesity, but the level of complications they have. I think this is an interesting discussion point, but I would like to go to the next point. And that in this first study, you used 12 females and 5 males. And so we already, and of widely different BMI, and it may be a purpose for the study, but I think that males and females, particularly regarding obesity, are highly different. So wouldn't it, you know, you now have a big mix of different people, and does that actually not reduce to, so maybe less strong conclusions about your study? And really testing the methodology. So if you would have taken only males and of a homogeneous population, would it not be much better to really quantify and say whether the method works or not?
This is referring for the study in chapter 3. Yes. This is true indeed. The population is mixed, predominantly with females and a few males. Ideally, as described also in the appendix, we would like to have recruited more individuals also that they were males, and to also execute comparisons for that. This was not done. When it comes to BMI, most of the individuals were within normal weight. It was only one individual that it was with obesity. We have executed the statistical analysis for with and without, and we didn't see important differences, and we thought of including as it was included in the study, as we could not predict that we could not complete the study, with including more individuals. The thing now, when it comes that the primary aim for the study was to see that indeed it can really measure these differences. We didn't know if it could be a method that definitely in the next should be follow-ups to include and examine if this is feasible to be done, or to detect differences between males, females, and different body fat distributions.
But then I would like to go to the question that was asked by the first opponent, and that is, so if I look at these adipose tissue depots, the femoral subcutaneous really is that is more blood flow to supply the adipose tissue. If I look at the femoral, especially the internal, it's often around blood vessels and these type of things. So you have a completely different anatomy. So does that really prove that you have functional differences in adipose tissue, or do you just measure the way the tissue is organized?
So you mean the question is about the anatomy and how we can be sure that indeed we're measuring blood vessels, that they are draining particular blood flow and blood tissue, fat tissue of the particular fat depot. This has been shown before, that the veins that we were examining from studies, for instance, from the groups in Oxford, that in particular for the branches in the femoral fat depot that indeed are predominantly draining the subcutaneous femoral fat depot, so that this is representative of this fat depot. And next, also for many studies that it has been much more, let's say, examining the abdominal fat depot, that the veins that we also choose to measure, to execute the measurements, are really representative of the drainage system of the subcutaneous adipose tissue.
Okay. Thank you. You clarify it a lot. Thank you very much. I would now like to go to the measurement of mitochondrial respiration in particular, and actually all the mitochondrial parameters. So I could not really find how you normalize your samples, but I understood that most of the samples were normalized based on BCA assay and so on total cellular protein.
Yes, that's true. We have measured the amount of proteins with help of technicians. I have not done it myself. We have measured the protein per well of cells after we have executed the dynamic experiments, and then we normalize based on that.
Yeah, but if I look at your pictures, for example, the 5.1 panel A or H, it doesn't really matter. I don't see a lot of differences in the patterns of respiration. What I see is differences in the levels of all the parameters, all the runs that you do. So I wonder whether, is it not because you test it on the amount of protein and not on the mitochondria, and so that you actually, by normalizing, you change all the parameters, and mitochondrial function is perfect in all the tissues, because otherwise you would have maybe seen differences between maximal, etc.
The thing is that indeed we have also executed the statistics and we have visualized that without the normalization, and the pattern was the same, but we thought with consulting also experts from our collaborators that it was maybe better to use the normalization units. That this is.
Yeah. But I agree that it is better to normalize, but I think I would normalize maybe on the number of cells, or maybe even on the number of mitochondria in some way, but in the number of cells would be easy. Maybe even viable cells. So did you consider that, and could that not explain why you see differences in these respiration curves?
This is indeed true. Maybe different ways of normalization could result to changing the visualization of the results. Yes.
So, because I think because it could mean that you basically also have a decrease in number of mitochondria per amount of protein, but not necessarily in mitochondrial function, which would be quite a different conclusion.
Yes, that's correct.
Okay. Then you also say that you look at differences between the cells and the tissue, and you basically say, okay, there's no differences, so the cells are representative for the tissues. But if I, for example, look at the mitochondrial DNA measurements, in one case they are significantly different, and the others they are not. So are they really the same?
The question is if the cells are the same.
You say, okay, I can nicely compare it because I don't see statistical significance. But in one case, you see a difference between the abdominal and the femoral in the case of the cells, but not in the case of the adipose tissue.
Yes, that's true. I think that was in the comparisons, in the subgroup comparisons per BMI group, that was either in the individuals with normal weight or obesity. But the thing is, when we compare definitely biopsies, it's not only the fat cells that we have there. There could be that we have an infiltration of more immune cells that are more active, and that's indeed not purely adipocytes in the tissue, also progenitors or endothelial cells. And we just examine a fraction of the tissue, which is of course, of bigger maybe metabolic index, which is the fat cells, but it's not definitely representing the situation in the fat tissue.
Okay. I think I have to stop in view of time. I'm not 100% sure.
You have time for a short question.
Then maybe one short question. This is related to something else, basically to inflammation. And you correctly state that you see changes in markers of inflammation. But how sure are you that you measure inflammation?
Excuse me, could you, I didn't listen to that.
So you state that you measure, you see changes in markers for inflammation, but how sure are you that you really see changes in inflammation in the tissue, and what could you do to look at that?
This is something that, this is true, of course, because these are indicators, not truly could represent what is happening. A better way of examining that, and that was something that we wanted to do, could be to use the biopsies and examine the population of the immune cells that they are recruited or existing in the fat tissue, and examine, for instance, that could be with fax analysis. It was in our intention to do that in a subgroup of the population, but it was not achieved. I think this could be a better way of determining, indeed, if more pro-inflammatory immune cells are infiltrating the fat tissue.
Okay, thank you very much for your answers. I give the word back to the Director.
Thank you. The opposition will be continued by Dr. Michailidou, Associate Professor in Metabolic Health at the Nottingham Trent University in the United Kingdom, and she was also member of the assessment committee, and thank you for participation to this ceremony.
Thank you very much, Mr. Prorector. Mr. Candidate, thank you very much for your presentation. I really enjoyed reading your thesis. It's really well written. I think I want to go back a little bit on the oxygenation availability in adipose tissue, especially with regards to your results. Can you elaborate on the differences between the oxygenation availability or levels in the peripheral versus abdominal, let's say, oxygenation levels, and how does this affect really function, in particular lipolytic capacity of different adipocytes?
Dear esteemed woman, thank you very much also for your kind words and for the question. So if I understood well, the question is if we know that if there are differences when it comes to oxygen supply between the two fat depots, and for instance, based on, as far as I know, there is only one previous study that has examined this, the subcutaneous abdominal and femoral fat depot, and it has shown that indeed there are differences in oxygen levels within the fat tissue. This is not necessarily representative of the oxygen supply, could be of course also of the oxygen demands. Theoretically though, the oxygen amount in the arterial blood vessels should be the same. I mean, when it comes to the amount of oxygen in the tissue, then what could change is the arterioles, I mean, the dynamic changes that allow higher or lower perfusion of the tissue. But I'm not aware of a direct study that has, apart from the one that was previously done from the lab, to show other differences. And in my study, I have measured with an indirect way the use of oxygen in the tissue, so not the supply to the tissue directly.
And what was your finding in terms of lipolytic capacity? Was the difference in your depots?
I have not executed.
Have you looked at non-esterified fatty acids?
Sorry. Yeah, so indeed, there were some differences that when we were comparing between the two fat depots, it was slightly higher in the abdominal, if I remember also correctly. This, is it possible to mention maybe the results or the figure in particular? Because, no.
I can't remember.
So you mean for when it comes to the results for the non-esterified fatty acid measurements in that was slightly higher in the abdominal, while we have not executed in this subgroup comparisons statistical analysis. Indeed, we have seen that to be because of the low number of paired samples due to the difficulties of the study and the recruitment. We have seen that indeed it was slightly higher in the abdominal fat depot, and this would represent that under fasting conditions that it's slightly a more dynamic depot, that it's providing in everyday life the substrate for other tissues, while it's accommodating for a more chronic, let's say, perspective, the fat in the femoral fat depot. So that could be an explanation for the higher NEFA.
Thank you very much. I guess I want to go back to this debate we've got about in human studies why there is a discrepancy between different labs. Some people find that in humans there is high oxygen availability in adipose tissue, and others find low oxygen availability in adipose tissue with obesity. So what is your take on this? How can we actually unify our way of measuring really oxygen availability so we all kind of have come to a consensus about this?
Well, that's indeed a very interesting point, as we don't still have a unanimous conclusion on this. I would like to start by maybe explaining the discrepancies. I don't think that is necessarily the methodology, because if a methodology is not working properly, for instance, we could not see the differences in different populations within BMIs with other methodology. Maybe there is to one extent. I think it's a matter of when we examine an individual, meaning in the natural history of a disease, that in earlier stages, maybe we indeed see hypoxia because it's necessary to drive and it's, let's say, a part of the remodeling of the tissue, so it's necessary to trigger angiogenesis, positive inflammation to do that remodeling. Further, going to a period of more stabilized after years, maybe we see here an equilibrium, and we see the hyperoxia or the higher levels. And then after more years, after a point, maybe we still see again to going down, like more hypoxic tissue. And if we also consider different BMI groups, those individuals with severe obesity to have even lower than the ones with moderate obesity.
Okay, I think I agree with the fact that different populations will give you different oxygen availability. I guess my other more generic question is in two parts. So if you put your clinician hat on first and tell me from your thesis, whatever you have found, if you were going back to the clinic and you had patients with abdominal versus femoral obesity, adiposity, is anything of your findings would make you think and change your approach of how you would treat these people?
Definitely, there are some things that we could go on with them and further examine them. We could use observations, for instance, with biomarkers that they are linked to a body fat distribution, and we think if we can use them for a predictor of disease outcomes. For instance, if we know that a specific biomarker is linked to lower fat accumulation, this theoretically is protective for an individual. We could potentially predict that different ways of therapeutic avenues of losing weight. No, sorry, there are two things that I want to say. That on the one, maybe we could predict based on the biomarker if there is an increased risk for developing disease. Another part could be that based on body fat distribution and a potentially biomarker, either inflammatory factor or mitochondrial or oxidative signature, to predict the responsiveness to a specific treatment option, and maybe be more personalized when it comes to that in everyday clinical life.
And how far?
Because of timing, I believe we have to move forward.
Okay, thank you very
with the
much.
next opponent. Thank you. That will be Dr. Hardy, Associate Professor in Steroid Metabolism and Signaling at University of Birmingham. And Dr. Hardy was also a member of the assessment committee.
Thank you very much. It's a really beautiful body of work, Ioannis. I very much enjoyed reading it. I'd like to start just briefly by saying, have you come across the work of Hennecker in the last year? It's a Lancet paper, very much focused on omics analysis of visceral tissues at different sites and across obesity.
Dear esteemed opponent, thank you for your question and the kind words. I haven't, no, I'm not aware of this work
I mean, I would. It's a good read anyway to have a look through in relationship to your work. I guess they use a number of transcriptomics platforms to look at these different sites, and they particularly focus on mitochondrial and inflammatory readouts at different sites with an LCMS and a transcriptomic approach. And I guess there's two things that I would like to consider briefly. One of them is that adipocyte size has quite a big impact on the metabolic profile and inflammatory profile of those cells. Based on your observations of different adipocyte size, what kind of changes might you have expected to see? Should you have looked at different sizes of adipocyte, and what kind of things might you have expected to see there or predicted to have seen?
This is, you mean when it comes to the laboratory in vitro work, if we could see, yes. That's indeed interesting because, as I said, we could expect that hypertrophic cells, bigger cells, theoretically should have been linked with a more pro-inflammatory profile, and theoretically we could see that based on visual inspection, we would not detect big difference in the size of the cells in the ones that we have used in the laboratory. We could assume that maybe indeed the ones for instance, deriving from individuals with obesity, because we see the difference in the fat cell depots, the difference in the fat cells in the biopsies. But this is something different. It's not translated to the laboratory conditions. I think that pretty much the cells are of the same size, so that's why we don't see potentially the expected difference, if this is linked indeed to the size of the cells.
Thank you. And in some of the larger cells, they had a very unique pro-inflammatory profile. Some of the things that came up were things that you've identified, some of the chemokines and pro-inflammatory drivers. So I guess from looking at your study, what's your understanding of how hypoxia, normoxia, and hyperoxia impact on macrophage polarization and inflammatory status?
So indeed, we have not directly, of course, examine that, so it's a bit of a complex question to answer.
Don't worry, I mean, I guess a simpler way of putting it would be, in hypoxia, there's quite a bit of evidence to suggest you get an anti-inflammatory pro-resolving macrophage phenotype, which might be important in vivo in your tissues, and the opposite's true with normoxia and hyperoxia. You get the more pro-inflammatory, classically activated macrophage phenotype. Given those sort of observations where you're looking at those different depots, what do you think the hyperoxia inflammatory activation of the macrophage might mean for your observations in the different adipose sites based on their oxygenation and blood flow?
Okay, yes, that's indeed. It could be speculated that for the observation that we have seen, the big difference is when it comes to the pro-inflammatory adipokines, it goes for the fat cells that they were deriving from individuals with obesity. And it could be said, like, as we have the idea of a memory of the immune memory and the interaction of the cells that are infiltrating fat tissue, maybe this is also inherent in a way, a memory of origin, memory of conditions of the cells that they are living in these conditions that they have lived before, like also the fat cells. And potentially this environment triggers responses that they are mimicking these conditions that they were there before during this, let's say, unfavorable for the cells time in the fat tissue. So this could be one part, like indeed interaction from the memory of the immune system and then the epigenetically memory in the fat cells to be linked with levels.
Thank you. And then could you have modeled that in vitro?
I'm not aware of 100% of that. I could speculate that maybe we could culture fat cells and macrophages and see the interaction in several also levels of oxygen.
I guess it would be really interesting. And a sort of a final line of questioning from me, when it comes to sort of translational interventions and where you would come from this, I guess you've identified changes in mitochondrial function potentially across sites, and you've also got slight differences in inflammation. But can you translationally target those? And are you familiar of any studies? I mean, I'm coming at this from the inflammatory aspect, but how easy is it to target inflammation in obesity to improve outcomes based, and has your study provided additional insights?
So indeed, I think there are a few studies that they have indeed in individuals with obesity or also diabetes that they have tried in the whole body to target inflammation, and it seemed at some point that it is positive, but I think there are conflicting results. Like when it comes to depot differences in inflammation, I'm not sure that it could be feasible to locally target this, so it could be in a whole body level. Potentially could have beneficial effects if we could reduce inflammation based on some studies that they have examined that from a perspective, for example, for insulin resistance.
And based on your study, should we be targeting the adipocyte or an immune subset?
I mean, I'm not quite sure, to be honest, which one of them, but if we target a receptor or an either that exists in both cells, could be either, or could be more specific when it comes to, like for instance, in other diseases, that to be a very specific target against subpopulations of immune cells so that they don't trigger further inflammation.
I'm very happy with your answers, and I'd like to hand the word back to the director. Thank you.
Thank you.
Thank you.
The opposition will be continued by Dr. Van Grevenbroek, Associate Professor Internal Medicine at Maastricht University.
Thank you. Well, Mr. Candidate, let me as last person in the row also congratulate you with this very nice thesis and also congratulate the promotion team. Yeah, you did good job also during the COVID lockdown, which must have been quite challenging given the, well, human aspects of the work that you've been doing. I would like to discuss with you some observations you did in chapter 4. In that chapter, you measured a lot of things in a lot of different depots and also the in vitro cells. And I've been looking at, okay, how do you connect all those types of information that you get? And for this, I try to focus on MCP1, and which is, as you also say, a pro-inflammatory molecule, and it's a monocyte chemoattractant protein, and in animal studies, it attracts monocytes to the adipose tissue. Now, in your data, I saw that it is slightly increased in the arterial blood, not significant. The fractional secretion rate was higher from the abdominal than from the femoral depots. But then it got a little bit more difficult because at the same time, you showed that the expression of MCP1 was higher in the femoral adipose tissue, and also that in your in vitro adipocytes, the MCP secretion was higher from the femoral adipose tissue. So looking from a naive point of view, you could say, okay, so then the femoral tissue attracts more monocytes. Is that pro-inflammatory or not? So how do you see that?
Yes, opponent, thank you very much also for the question and the kind words. This is true that it seems also for us that could be kind of surprising, this observation as you have described. Yes, indeed, in the fat depots in human studies, we see only these differences, and then we could consider that in the femoral fat depot, it's something happening. It could be that this is representative of a positive degree of inflammation, represented more remodeling in the fat tissue of the femoral fat depot. When it comes for the discrepancies, at least could be also that other local factors are not, could be acting as a paracrine and not into the circulation, potentially. The bigger discrepancy maybe when it comes from the biopsies to the cells, it could be claimed that indeed other cells that were regulating in ways that we cannot mimic in the laboratory conditions. That's why we could have these discrepancies.
Yeah, so how would you continue with that? So what do you think would be the line to continue with these observations you got so far?
Yeah. In particular for MCP1, because I've said it's an indicator that usually we expect more macrophages, it should be a follow-up study to examine potentially in this population or other population the presence of which kind of cells they are indeed present in the fat tissue depots, and also measure again this and see if it corresponds to the presence of macrophages in the tissue.
Yeah, I can see that. I could also get that you might want to try to really get the adipocytes as they are in the adipose tissue somehow to see how they are expressing this MCP1. And a little bit related to that, the fact that you did quite a lot, and so you have a lot of data, but you also have relatively small groups, and you refer to that in the methodological consideration that you say because this is an explorative setup, we did not do power calculation, and we decided not to adjust for multiple testing. And then for the power calculation, you relate on a previous study. And now I looked up that study, and that was a intervention. They did something with the infusion of hydrocortisone, I don't know exactly. So how do you translate those numbers from an intervention to your observational explorative study? So how did you come up with the number that you eventually included in your analyses?
Yes, this is also based on another example of this type of studies. Maybe, of course, it seems from a clinician point of view that we see randomized controlled trials with much bigger numbers. This is a small study compared to this. But when it comes to comparing other physiological studies, even though it seems that it's lower, it's representative of them. The thing is, of course, that I can continue maybe finish. Yeah. I can finish, or yes. So, or I can finish in the reception. Maybe I don't know if it's not enough.
Okay. Thank you. Ioannis Lempesis, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberation and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Ioannis Lempesis, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Blaak is authorized to confirm upon you this academic distinction in accordance with the Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principle of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Ioannis Lempesis, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. So congratulations, Ioannis. You did a very good job for the second time. And I now would like to pass the word to your supervisor, Gijs Goossens, who will do the Laudatio.
Dear Dr. Lempesis, dear Ioannis, congratulations on reaching this very important milestone. And it's a good habit to look back and reflect on the years that we've worked together during your PhD journey. And of course, I will also do this on behalf of Konstantinos and Ellen. So it all started back in 2017 with a call for a joint PhD project between the University of Birmingham and Maastricht University, aiming to stimulate collaboration between the two research institutes. And I decided to contact Konstantinos to discuss the possibility to work together to investigate depot differences in adipose tissue, metabolism and inflammation. And Konstantinos had been trained in Oxford, where he worked with Keith Frayne and Frederick Karp, so he was really, I would say, well acquainted to perform all these metabolic flux studies across adipose tissue. And combining that with biopsy work and in vitro experiments, we thought this would be really a nice synergy between both labs. Unfortunately, our research proposal was granted, and with some additional funding from the European Foundation for the Study of Diabetes, we could perform all the experiments we had in mind. So then we start looking for a PhD student. A PhD student who would be able to perform in vivo studies in humans, preferably also with some affinity for cell culture work. And of course, the candidate needed to be willing to work in both institutes, because that was also one aspect of the joint PhD program, to get acquainted and work in different academic environments. And the interview with you took place on October 9th, 2017, almost 6 years ago. And you had a medical background, spent several months at Imperial College in London, where you had been involved in several clinical studies in humans. You were enthusiastic about working in both institutes, and you were also keen to learn more about adipose tissue biology and obesity. So therefore, we thought you would be a very good candidate and decided to appoint you as a PhD student on the project. And you started working on the project in January 2018. And 2 months later, we already submitted the project for ethical approval. And while waiting for ethical approval, you already started to write a review article. That is actually chapter 2 of your thesis, and it was accepted quite rapidly thereafter. And 6 months later, we obtained the medical ethical approval and could start recruiting the volunteers. But then, as you already alluded upon, xenon, which was needed for the blood flow measurements, was not available anymore. So there was a production stop by the manufacturer. So that was a disappointment, and we had to look for an alternative. So we also needed to submit an amendment to the Medical Ethical Committee, which again took quite some time. And I think you were quite frustrated about that process. It caused quite some delay. But in the meantime, you finished that review article and also initiated the study to determine whether we could use Doppler ultrasound as an alternative technique to measure blood flow in adipose tissue. And that is, of course, chapter 3 of your thesis, which was also published shortly after completion of the study. But recruitment of the study participants for the main study really took much more time than anticipated, likely because the measurements we aimed to do were rather invasive, including both blood sampling from the radial artery and different veins in the fat tissue, collecting biopsies and so on. So we tried many ways to recruit the study participants, from advertisements to recruitment via GP networks and so on, but all things were not just moving forward as you would have hoped, actually, with respect to recruitment. So then we decided that some additional study participants that we needed to complete the study could be measured at the University of Birmingham, I think with the help of several great colleagues over there, including the research nurses. And after about 20 months in Birmingham, that you would already come over to Maastricht to work on the other aspects of the project, namely the in vitro experiments, analysis of the tissue biopsies, data analysis, manuscript writing, and so on. So you learned how to culture adipocytes and how to handle the cells. And I must say that took quite some effort, given your medical background. But I think in the end, you managed and you started the analysis of all the blood samples and the biopsies that we had collected in Birmingham. But then when all these in vitro experiments were just up and running, we encountered the corona pandemic in March 2020. And a difficult period followed. Also a difficult period for you, with limited access to the lab and also some motivational personal issues that occurred at that time. So you experienced extreme stress due to the uncertainty of the situation and also worries about your PhD timeline. But I think with the excellent support in the lab from multiple colleagues, including our technicians, Nicole and Yvonne, I also see that Yvonne is here today, we managed to complete all the work. And although it was not easy for you to deal with the situation, I think really, Ioannis, you demonstrated perseverance and were committed to successfully complete the study's statistical analysis and manuscript writing. So you finished your PhD project with only slight delay and had the opportunity to present some of the key findings during an oral presentation at the EASD in Stockholm last year. And about 2 months later, you successfully defended your thesis during the Viva in Birmingham. But to obtain a double doctorate degree, you still needed to rewrite and change certain parts of your thesis to comply both to the rules and regulations of both universities. It required extra work, but you did it, and I think you managed. And although we talked a lot about science during our meetings, we could not help noticing that you have a special fondness for poetry, literature, and the arts. And therefore, I would like to recite a poem by the Dutch comedian, singer, painter, and poet Toon Hermans, who was born more than a century ago in Sittard, actually just 15 minutes away from here. And the title of this short poem is called Day. And it's about the fact that not every day can be perfect, but that one needs to accept that there can be bad days as well, and that it is okay to feel down so now and then. And I've tried to translate this into English for you. It's a short poem. Day. There is one day every day. Every day is unique. One is a pit and the other a peak. Give every day its own side. One is open, the other tight. One is perfect and the other upset. You never make a pit a peak of a day. I'll give it to you, you can read it later on, but I hope you'll think about this poem so now and then. So it's okay to feel down now and then. I think that's also important to mention. So Ioannis, it has been quite a journey. Working at 2 research institutes, getting familiar with all the research methods, hindered by the corona pandemic, but you can be very, very proud of what you've achieved. A very nice thesis, 4 chapters already published in well-respected international scientific journals, and a final manuscript currently in revision. And you have the ambition to pursue a career as a clinician working in the field of obesity and diabetes, combining your clinical work with research. And you have a clear wish to do this in the US. And therefore, I'm very happy for you that you have obtained a position as a postdoctoral fellow in the lab of Frank Shear at Harvard Medical School in Boston, and I really wish you all the best. And finally, I would also like to take the opportunity to thank the members of the Thesis Assessment Committee for reviewing Ioannis' thesis, and a special word of thanks to Drs. Abtarani, Zoe, Michailidou, Rowan Hardy, and also Jaap Keijer for your efforts traveling to Maastricht to be part of the ceremony today. So Ioannis, thank you for the collaboration over the past few years. It's now time to relax and celebrate this achievement, and congratulations once again also on behalf of Konstantinos and Ellen, and I would of course like to include your family and friends with these congratulations, so thank you.
Thank you.
Dear Dr. Lempesis, also on behalf of the Maastricht University, I will congratulate you with this new degree of doctor. The University, Maastricht, and the Board of Deans wish you all the best in your future academic and clinical career, starting at Harvard, just heard. And personally, I hope that besides the lyrics, the poem of Toon Hermans, maybe also the lyrics of Thomas Eliot may inspire you during your future career. Hereby, I close this academic ceremony. Before congratulating the new doctor, we will take a picture with the new doctor on the stairs in the central hall.